As we continue to take this information beyond V1, we are going to continue to talk about the what pathway through the ventral root, through the temporal lobes. And I'll come back to our lollipop neuron just to say that, again, we're getting beyond just seeing features. We're putting those features back together into objects. And here, Tanaka et al. Found an area that is still with kind of simple objects, just putting a couple features together, right, as we get to our lollipop neuron. So as we get to more complicated putting those features together, what they discovered actually was modularity, that we have these what they called modules, or these specific brain regions that are specialized to respond to specific types of stimuli or stimuli that perform specific functions. So we're going to end up talking about the region that responds to faces, or a region that responds to places and things that help us to navigate in our environment. But they have found a region or regions that respond to animals or to tools, so performing some function. And each of these specific areas is called a module. I will continue to talk about distributed processing, as it's not just that one module that is responding to something like a face, that we're going to have activity distributed throughout the brain. Much of the research that looks at this kind of modularity has used brain imaging, and particularly that fMRI, that functional magnetic resonance imaging, making it possible to create pictures of precise location of brain activity. So again, one of the regions that was found is a region that responds to faces. This work was done more early on by Rolls and Topi, where they were measuring from the infratemporal cortex of monkeys. And you can see, looking in this region over on the figure, that there was activity when looking at faces, or the stimuli in the top row. And then there was really very little, almost no activity. So it actually looks like a little bit of inhibition to places, flowers, other things that are not faces. The Sao et al., they took more different specific categories to look at exactly what's happening with different categories. And again, we can see the neurons are responding to faces, but not to bodies, fruits, gadgets, hands, or scrambled pictures. And in 1997 is when Nancy Kanwisher et al. Used fMRI to find the fusiform face area, so the region in humans that responds best to faces. And you can see that area of IT, infratemporal cortex, in monkeys. They're finding these little various regions, but they're basically finding this is IT, the infratemporal cortex. If we turn our brain upside down, that fusiform face area is actually on the right side in the infro, so infro being at the lower part of the temporal cortex. We also see neuropsychological evidence for this brain region being important to facial recognition, because we have people who have damage to this area of the temporal lobe, the fusiform face area, and they end up having what's called prosopagnosia. So this difficulty in recognizing the faces of familiar people. So this is just additional evidence that this region is specialized for the perception of faces. And I do have a couple of vignettes or stories for talking about prosopagnosia, and I'll use the one that's a little bit funny, even though it's kind of not, if you really think about what all this means to somebody. So this was somebody who had damage from a stroke and damaged the fusiform face area pretty severely. He's talking to his wife in a restaurant, and he's saying, "Hey, that guy's looking at me. He's looking at me funny." And his wife says, "Don't, just ignore him." And he says, "He's giving me a dirty look. Every time I look over, he's looking at me and giving me a dirty look." And she says, "Just if you don't ignore him, it's like he's not there. Just completely don't pay any attention." He got more and more upset until finally he started to stand up. He's like, "I'm going to have to talk to this guy. He's just staring at me." and giving me a dirty look. And he starts to stand up, and he's like, oh my goodness, he's getting up too. And his wife looks behind her in the mirror, and she goes, that's you. So this is how profoundly damaged this region can be, that you wouldn't recognize your own face. And I like to include this picture for a number of reasons. First, pictures are fun, but also it shows how specific some of this damage can be, and that we can also have regions that are spared when we have some kind of damage. And so we talked about visual. It also brings us back to visual object agnosia. So we talked about visual object agnosia actually when we talked about Mr. P, the man who mistook his wife for a hat, when we talked about the perceptual process. And I said we really need to make sure that we distinguish between perception and recognition, because I can perceive the world, I can describe the world, or he could, right? He could perceive the world and he could describe the world. But putting that together into recognizing the actual objects from the world, that takes this area of the temporal lobes. And so this picture is shown because they had a patient who could recognize the face. So that fusiform gyrus, that fusiform face area, was intact, but not the flowers, fruits, and vegetables and everything that makes up the face. So they had a profound kind of visual object agnosia, but the fusiform face area was preserved. And just to go through two more of these regions, again, they found your book goes through quite a few different regions, and actually looking at what's happening in particular films of and scenes and so forth of what the brain is responding to. But two that were discovered relatively early on and that are quite important are the parahippocampal place area. So that PPA is the parahippocampal place area. It's around the hippocampus, and they talk about it as helping us to recognize places. But if you can notice the top stimuli that it is responding to, well, that one that's, so there's a mountain looking thing, there's a house looking thing. The next one over is a corner, right, of a room, an empty corner. So, and the next one over is, it almost looks like little, almost like Lego houses or something. So anything that can suggest that we might use it to navigate appears to stimulate or activate this parahippocampal place area. But if we look at rockets or scrambles or single Lego pieces or whatever those are, they do not activate it in the same way. And then the bottom one is the extrastriate body area, or the EBA, as it is labeled there. And you can see that it's going to respond to other body regions, but not to hammers, not to scrambles, and not to faces. We have a different region of the brain that's responding specifically to faces. But other body areas, whole bodies, stick figures, shadows, are extrastriate body area. But as I stated at the beginning, this is truly distributed processing. If we look at the processing for, let's say, houses, so if you look at B right above it in houses, actually I'm going to use faces. I like to use faces. At the activity for faces, it really is all over the brain. They're showing us two successive coronal slices of the brain, and there's activity all over the brain. There's particular activity. If you notice, the yellow is the highest activity there on the right side in the infratemporal region of what we call the fusiform face area. And so remember, what we're doing when we're doing an fMRI study is we are using this comparison, right, or this relative activation. And so if we look at the picture in A, where I've subtracted out the other objects, and I'm looking at, this is what's responding to faces. A is if we look at the red and look at faces, we see that fusiform face area appears to be quite specific to faces, but the brain is active all around for faces, because when we subtract that out from looking at something like houses, then we're going to see a different pattern of where we are responding more to houses. But there is distributed activity. And one example would be the brain responding to a face. So that information is coming in right through the lateral geniculate nucleus, going to the visual cortex. So those regions of the brain are clearly active. We also respond to emotional aspects of a face in the amygdala, where someone is looking, or how the parts of the face move. All of this kind of biological information, biological motion information that tells us like joint attention and things like that. The superior temporal sulcus is active for that. I'm going to go ahead and go through whether the face is familiar. So the fusiform face area tells us, and our hippocampus tells us, is this familiar? And then your author has how attractive a face is, and he puts that in the prefrontal cortex. I kind of feel like in my prefrontal cortex, I have a bit more going on, like things like, do I owe this person some work? You know, would I want to work with this person? Do we have a dinner plan with this person? All of these more, I think, of higher processing being a little bit more than just how attractive a face is. This is not to, and here's a nice table of all of this. This is not to give you a lot to memorize. You don't need to remember this. Hey, or here, these are all the regions that are active when we're looking at faces. The point of this is to show this is very distributed, right? We're seeing the maximal activity in the fusiform face area, but we're seeing activity all over the brain in response to faces. And now we're at a point where we are representing objects instead of just features in the environment. And we might ask ourselves, how does neural firing represent various characteristics of the environment? And there are a number of possible explanations for how this happens. One would be specificity coding. And I'm going to talk about specificity coding versus population or distributed coding. This argument is going to come back for all of our sensory modalities. It's there for all of our sensory modalities, but it's going to come back specifically. It ends up coming back for sound localization as well as for taste, and as far as within the textbook. But this is an argument, or this is a line of research for every sensory modality. There are many questions of how are we representing this sensory information with neural firing. And one possible explanation is specificity coding, where we would say the representation of a particular object in the environment, in the case of vision, is going to be represented by the firing of a specific neuron that is tuned to respond to that object. And this is where we get this idea of grandmother cells. And there are people who believed, especially in the 60s, that we had these grandmother cells, neurons that responded only to that specific stimulus of my grandmother. And so this neuron would respond to the image of your grandmother, the concept of grandmothers in general, the name of your grandmother, all of these things that get locked together into grandmother. And your book has this kind of visual representation, which I'm going to steal an old one. This would mean that for faces, I have a neuron that responds to Bill, a different neuron that responds for Mary and not to Bill or Raphael, and then I have a separate neuron responds to Raphael but not to Bill or Mary. I do ask this question, and it is something to think about, right? So pause, reflect, think about this. So do these neurons really exist? Do we think they really exist? And why is it unreasonable? Why would it be unreasonable for our entire nervous system to work this way, for us to have specificity coding for something like faces? And to leave that bit of silence. I know it's awkward and weird in a video, but I do think it's important that even if you don't decide to pause and stop, that there is a pause, a stop, where you think about that question. So what are our options? One option is population coding. So what this would mean is that a particular object is represented by the pattern of firing across a population of neurons. So this way we don't need these specialized neurons for some specific, each specific object that I've ever experienced in my life. Another option is sparse coding. So we have a relatively small number of neurons, and it's their pattern of activation of firing that is going to tell us this is the object that I'm looking at. And this is probably responsible for the Quiroga results that your author mentions, that there have now been several studies done by Quiroga and colleagues where what they did was they were doing single cell recording from, I want to say it was the hippocampus, not the fusiform face area, but the hippocampus, while people were about to undergo surgery for epilepsy. So their brain is exposed and they can go in and do this kind of single cell recording. And what they did was they showed them pictures of faces of famous people like Halle Berry and Jennifer Aniston. They showed them other faces as well, so they showed them many faces. Okay, what they found was a Halle Berry neuron, basically. They found a neuron that responded to Halle Berry, her name, her in her Catwoman costume, her in different poses. So this appeared to be like a Halle Berry neuron. And there were people who interpreted this as, oh, this looks like, look, there's that idea of a grandmother cell is true. We have a neuron for Halle Berry. But Quiroga et al.'s, their argument against this was that they are recording from a region that has billions of neurons, right? And they have chosen certain neurons to record from. They've chosen certain spaces to show. And this would be like finding a needle in a huge stack of needles by accident. So that didn't make sense to them that this would be evidence for specificity coding, but it does appear to be evidence against population coding, because otherwise we'd be seeing Halle Berry also responds to Jennifer Aniston and maybe other females, maybe other actresses. What's interesting is the Jennifer Aniston neuron that they recorded from also responded to Lisa Kudrow. And if you know your Friends people, they're both females, right? Female actresses that were in the show Friends. And so do we have a Friends neuron? Well, it's probably, they say most likely it's not population coding, but it's probably a sparse coding. So there's probably some small or relatively small population of neurons, and it's the pattern of firing of those neurons that tell me that this is Halle Berry. And so we see activation of this neuron for Jennifer Aniston and for Lisa Kudrow, but there's going to be some amount of difference, right, in that pattern when we look across that, again, relatively small population of neurons. The book doesn't completely distinguish between sparse coding versus distributed or population coding the way in a figure, but I did want to have another figure to show that what this would mean is that we have these three neurons that we're looking at, and this neuron 1 responds really strongly to Bill, neuron 2 and 3 not so much, maybe just a little. For Mary, it's neurons 2 and 3 that respond really strongly, and 1 not so much. For Raphael, maybe it's 1 and 3, and 2 maybe some. And then we can add other people, and they're going to have their own pattern of activation across those neurons. And so 3 makes it look extremely sparse. The question here then is, so to distinguish this out, of how large is this group of neurons? If it's a really large group of neurons, we're going to call it population coding. If it's a relatively small group of neurons, which again is what Quiroga et al. Suggested, really explained their results, we're going to call that sparse coding. So as a summary, putting together all of that specificity versus population coding, or in this case, what they found with sparse coding, is this is really an answer to, or this is a line of research examining this question of how are we representing objects from our world, or perceptual experiences with neural firing. So our options here are specificity coding. A single neuron firing represents some specific object. In the case of objects, sparse coding, it's a relatively small group of neurons that has a particular pattern of neural firing. That's going to be what represents a specific object. And again, that appears to be what really explains well the Halle Berry neurons, or the Steve Carroll neurons. I always say his name wrong, so sorry about that if I just said to him. And then the population coding is another option, where it's a large group of neurons, so many neurons in the hippocampus. They have their particular pattern of firing, some of them more strongly, some of them more weakly, and that's going to represent a specific object.